Okay, kids, let's talk about how DNA replication works, and I'm going to zoom in the replication bubble, okay? So, I'm actually just going to do one side, alright? 이렇게 생겼어요. Okay, this is what the DNA looks like. It's one side. Let's say this is going from 5 prime to 3 prime. Okay, this is going from 5 prime, yep, to 3 prime. Okay, so this one's going this direction. Alright, understanding that DNA is anti-parallel from each other. One goes up, the other one goes down. One goes right, the other one goes left. Okay, understanding that, we can actually figure out how the new daughter strands can actually be formed. Okay, so first of all, this area right here, this is called a fork. Okay, where the road kind of like forks and divides. This is called a fork, and on the fork there's going to be an enzyme called helicase. Okay, and what helicase does, it unwinds DNA. It starts to unwind DNA going this direction. Okay, super easy? Super easy. Alright, well, this helicase is going this direction. Let's talk about how the daughter strands are actually going to be formed. Now, understanding that the parent strand down here is moving this direction, right? It's moving this direction. We have to make sure that the daughter strand is moving this direction. Good time? All right, so the daughter strand is moving this direction. Okay? So it's going from 5 prime to 3 prime. And who's the one who's actually creating the new daughter strand? There's an enzyme called DNA Polymerase 3. Okay. Yes. DNA polymerase 3 is the one that's actually going to start making complementary base pairs. Complementary base pairing.
That makes the daughter strand, right?
Yes, it's making the new daughter strand. Okay, the blue is the daughter strand. Okay, good. All right. So what about this top DNA? Okay, this top DNA, the parent strand is going this direction, which means that the daughter strand has to go the opposite direction, that of the helicase. So understanding that the helicase, okay, starts out here, when it unwinds, the DNA polymerase can go and make fragments like so. Okay, it opens up a little bit, you go back, and you make another fragment. You open up more, you go back, and it makes another fragment. So you see how we made lots of fragments? These fragments have a special name. They are called, exactly, Okazaki fragments. They're called Okazaki fragments. Super easy? Super easy. But if you look again, we are going from 5 prime to 3 prime. 5 prime to 3 prime. 5 prime to 3 prime. Okay. Now, same idea. Up there, DNA polymerase 3 is the one that's doing the complementary base pairing. Okie dokie? Now, if you guys go back, okay, and if you remember, this is a nucleotide, right? We have a phosphate, a 5-carbon sugar, and a nitrogenous base, right? Okay, and then we have another phosphate, 5-carbon sugar, and a nitrogenous base, right? Okay. Now, if you guys remember, in deoxyribose, assuming that this is deoxyribose, okay, let's say this is ribose. They're not supposed to bond to each other, but I'm going to show you guys that they do for a small little period of time. So, if you guys remember, this is one nucleotide, this is another